Hello friends, welcome back. So today we're going to start a new chapter which is biological classification chapter 2. I'm so sorry I have not covered chapter 1 which is the living world, but I will soon try to complete it and we'll make a good series over this. So yeah, without any ado, let's get into the video. And before that, if you are new to my channel, I'm Ujjwala and I make videos regarding cool stuff like neat syllabus oriented biology and also some fashion content videos, etc. And if you're interested, subscribe, like, share for more content. And yeah, without wasting any of your time, let's start the video. Yeah, this is chapter 2, biological classification, which we are going to deal today. So yeah, I'm going to explain you NCRT line by line. Since the dawn of civilization in the sense that since the starting of the civilization, it means knowing the knowledge of human minds become they were started realizing, they converted the how the early man have developed into such a great technological person. The civilization is kind of stuff related to that. There have been many attempts for classifying living organisms. So there is a very diverse classification, right? Inta mandi organisms, inni organisms ni, like there are like you can see I have four. It means imagine that I'm having pens with caps. They're really not organized well. They're just kept like this. And if I suddenly want to find the correct cap and pen, I would just pick this. This is not matched, right? It means I have not segregated them properly. Segregation in the sense grouping organisms or grouping anything, like grouping pens with the correct ones where they are originally belonged. In the sense ఈ కలర్ పెన్కి ఈ కలర్ బాడీ ఈ కలర్ నిబ్ ఉండేదే మ్యాచ్ అవుతుంది. సో ద ఇంక్ ఇస్ రెడ్ అండ్ ద గ్యాప్ ఇస్ రెడ్. So these are matched. The same way organisms also have to be placed to their right positions where they actually belong. That is what classification meant. So this is very easy chapter. Only thing you need to do is proper revision. That's the key to remember and solve the biology, especially biological classification. Totally NCRT is more than sufficient, and solve some PYQs and some MCQs as well. Only NCRT thorough revision is required, and also you don't need to have any extra material, nothing. Even your college material is also very much to these chapters, because these are only mug up kind of chapters. If you have good memory power, if you can remember the dates, if you can remember the scientist names, and if you can remember all the points very well, if you have good memory, then you can get sent. I am sure you can. So yeah, let's get into this. I hope you got some knowledge. Yeah, yes, that is what classification is. You need to have proper arrangement of organisms. It was done intensively, not using criteria that were scientific. So many of them were trying to classify organisms. Chaala mandi organize cheyali ani try chesaru, but that was not based on proper reason. Without a proper reason, they started classifying. It was only classification అనేది ఒక్క కారణం వల్ల, పర్టికులర్ రీజన్ వల్ల కాదు, ఓన్లీ ఫర్ దేర్ నీడ్, అంటే వాళ్ళ అవసరాల కొద్దీ వాళ్ళు క్లాసిఫై చేశారు. దట్ వాజ్ ద ఫస్ట్ క్లాసిఫికేషన్. సో ద క్వశ్చన్ కమ్స్, ఫర్ ఎగ్జాంపుల్, ఇఫ్ ఎ క్వశ్చన్ అరైజెస్ ఇన్ యువర్ ఎగ్జామ్, what was the reason for the first classification, and the options would be out of need, and it was plants and animals, and it will be Monera, Protista, plants, animals, fungi, and it will be three kingdom classification. But the reason behind the first classification was it was born out of need to use the organisms for our use, for food, shelter, clothing. We need to organize. It was only the first time the classification was only based on the needs of humans. Aristotle was the earliest to attempt the scientific basis of classification. He was the first person to classify the organisms. He used simple morphological characters to classify plants into trees, shrubs, herbs. Trees are long, very big. Shrubs are shorter, and they are just 10 inches up to the ground, and herbs are very small than shrubs. This is a descending order which is given here. It may be also asked in the examination. Morphology in the sense external appearances. There were no point of physiological or based on the future evolutionary relation based on classification was not there in the Aristotle, and also these were only gross morphological structures. Outer appearance బట్టి అతను క్లాసిఫై చేసినాడు. ఆయన ఒక పర్టికులర్ అంటే దానికి జస్ట్ అప్పుడు అవసరాలకు కావాల్సినంతలా జస్ట్ సీయింగ్ ది ఎక్స్టర్నల్, బయట ఆ చెట్టు పెద్దగా ఉందా చిన్నగా ఉందా ఇంకా చిన్నగా ఉందా, ఆర్ఎల్స్ అది చెట్లకు, ఆర్ఎల్స్ హీ డివైడెడ్ యానిమల్స్ ఇంటూ 2 గ్రూప్స్, రెడ్ బ్లడ్ అండ్ డిడ్ నాట్. అంటే యానిమల్స్ ఒకవేళ ప్లాంట్స్కి వస్తే సైజ్ ప్రకారం, యానిమల్స్కి వస్తే దానిలో రెడ్ కలర్ పిగ్మెంట్, ఒకవేళ బ్లడ్ రెడ్ ఉంటే అవి ఒక గ్రూప్, లేకుంటే ఇంకొక గ్రూప్. ఇలా 2 కింగ్డమ్స్ అని అరిస్టాటిల్ క్లాసిఫై చేశారు. బట్ దిస్ వాజ్ నాట్ సఫిషియంట్ అండ్ దిస్ వాజ్ డిస్ప్రూవ్డ్ బై లినేయస్ టైమ్ 2 కింగ్డమ్ సిస్టమ్ ఆఫ్ క్లాసిఫికేషన్, ప్లాంటే అండ్ అనిమలియా. ఇంకా నెక్స్ట్ classification was done by Linnaeus, and he divided the whole organisms into 2, plants and animals. And the system did not distinguish between, అంటే ఈ లైన్స్ అనేవి మనకు లినేయస్ క్లాసిఫికేషన్ యొక్క డిసడ్వాంటేజెస్ని చెప్తున్నాయి. దిస్ ఈజ్ వెరీ వెరీ ఇంపార్టెంట్. దిస్ సిస్టమ్ డిడ్ నాట్ డిస్టింగ్విష్ బిట్వీన్ యూకారియోటిక్స్ అండ్ ప్రోకారియోటిక్స్, యూనిసెల్యులార్ అండ్ మల్టీసెల్యులార్ ఆర్గానిజమ్స్, ఫోటోసింథటిక్ గ్రీన్ ఆల్గే, నాన్ ఫోటోసింథటిక్ ఫంగై. సో ఇవన్నిటికీ డిస్టింగ్విష్ చేయకుండా జస్ట్ అంటే సపోజ్ ఫంగై అండ్ ప్లాంట్స్ వేరే కదా, ఫంగైలో మనకు హెటిరోట్రోపిక్ న్యూట్రిషన్, ప్లాంట్స్లో మనకు ఆటోట్రోపిక్ న్యూట్రిషన్, బట్ అవన్నిటిని మర్జ్ చేసి ప్లాంట్స్లో వేయడం ఇట్స్ నాట్ ప్రాపర్ క్లాసిఫికేషన్. బట్ ఆ టైంలో ఇది చేయడం చాలా గ్రేట్, బట్ దట్ వాజ్ నాట్ ఎ కరెక్ట్ క్లాసిఫికేషన్. దట్స్ వై దిస్ వాజ్ ఆల్సో disproved. A classification of organisms into plants and animals was easily done and was easy to understand, but a large number of organisms did not fall into either category. అంటే చాలా మటుకు ఉన్న ఆర్గానిజమ్స్ అటు ప్లాంట్స్లో గ్రూప్ కాలేదు, ఇటు యానిమల్స్లో గ్రూప్ కాలేదు. హెన్స్ 2 కింగ్డమ్ క్లాసిఫికేషన్ యూజ్డ్ ఫర్ అ లాంగ్ టైమ్ వాజ్ ఇన్అడిక్వేట్. అంటే చాలా రోజుల వరకు ఇది ప్రాక్టీస్ చేసినా కూడా అది ఉపయోగం లేకుండా అయిపోయింది. ఎస్ రైట్. బిసైడ్స్ గ్రాస్ మార్ఫలాజికల్ structures, a need was left including other characteristics like cellular structure, నేచర్ ఆఫ్ సెల్ వాల్. అంటే సెల్యులార్ స్ట్రక్చర్ అంటే ఆ సెల్స్ ప్రోకారియోట్సా యూకారియోట్సా అని, ఇంకా నేచర్ ఆఫ్ సెల్ వాల్ అంటే అది సెల్యులోజ్ ఉందా నాన్ సెల్యులోజ్ ఉందా, కైటిన్ ఆర్ absence of cell wall, like that, or mode of nutrition, they are autotrophs or they are heterotrophs, parasites or saprophytes, and habitat, they are aquatic, they are marine, or they are freshwater, or they are terrestrial. Method of reproduction, sexual or vegetative. Evolutionary relationships, main వచ్చేసి evolutionary relationships అనమాట. సో ఇవన్నిటి కన్సిడర్ చేస్తే దట్ విల్ గివ్ ఎ ప్రాపర్ షేప్ ఫర్ ద క్లాసిఫికేషన్ ఎక్సెట్రా. Classification system for living organisms have hence undergone several changes over the time, though plants and animal kingdom have been a constant under all different types. This line, it is very very important. ఎన్ని సార్లు క్లాసిఫై చేసినా కానీ, ఎవరు క్లాసిఫై చేసినా కానీ 2 కింగ్డమ్స్ మాత్రం డిస్టర్బ్ కాలేదు. వాట్ ఆర్ దోస్? ప్లాంటే, అనిమలియా. వెరీ 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 ఇంపార్టెంట్. ఎంతమంది క్లాసిఫై చేసినా the 2 kingdoms which were undisturbed was plante, animalia. These 2 were undisturbed, no matter who classified it. The understanding of what group or organisms being included under these kingdoms was been changing. The number and nature of other kingdom have also been understood differently by different scientists over the time. ఒక్కొక్కరికి ఒక్కొక్క ఒపీనియన్ ఉంటుంది. కొందరు కొన్నిటిని ఒక కేటగిరీలో చేస్తే ఇంకొకరు ఇంకొక కేటగిరీ, ఇలా టైం టు టైం ఒక్కొక్క సెంచురీలో ఒక్కొక్కరు ఒక్కొక్క విధంగా క్లాసిఫై చేయడం అనేది జరిగింది. Characteristics of 5 kingdom classification. ఈ బాక్స్ దిస్ ఈజ్ డామ్ ఇంపార్టెంట్, చాలా అంటే చాలా ఇంపార్టెంట్. దిస్ షుడ్ బీ ఇన్ యువర్ మైండ్. ఒక షార్ట్ అనేది మీ ఒక స్క్రీన్ షాట్ లాగా మీ మైండ్లో ఫిక్స్ అయ్యి ఉండాల్సిందే. దట్స్ ఫర్ షూర్. వెరీ వెరీ ఇంపార్టెంట్ అండ్ కీ టు దిస్ ఈస్ అ కీ టు అన్లాక్ ది బయాలజికల్ క్లాసిఫికేషన్. ఐ వాంట్ టు సే this, though it appears very simple, you can say it is very easy to learn, but it is very important to keep it remember permanently in order to solve the MCQs in a quicker, less time taking. Okay, R.H. Whittaker in 1969 proposed a 5 kingdom classification. Who proposed a 5 kingdom classification? R.H. Whittaker. The kingdom defined by him were named Monera, Protista, Fungi, Plantae, Animalia. So 5 kingdom classification includes Monera, Protista, Fungi, Animalia and Plantae. These were the 5 kingdoms which were placed under R.H. Whittaker's classification. I hope you are getting what I am saying. Okay, now the main criteria of classification he used was cell structure, body organization, mode of nutrition, reproduction and phylogenetic relationships. So he included all the necessary things for classification which we have studied earlier, right? So The three domain system in this sense, another scientist, six kingdom classification, which was later after Whittaker it was done again. Six kingdom classification have been proposed and divided the Monera into two domains. అంటే ఇక్కడ మొనెరా ప్రొటిస్టా ఫంగై ప్లాంట్ ఎనిమలియా ఉంది కదా, ఈ మొనెరాని ఇంకా సబ్ డివైడ్ చేసి రెండు క్లాసెస్గా చేశారనమాట. Who did six kingdom classification అంటే Carl Woese, he did the six kingdom classification. He divided Monera into Archaebacteria and Eubacteria. That was the class subdivision of Monera. And have been proposed that divides the kingdom Monera into two domains, leaving the remaining eukaryotes in the third domain. So first three domains kada, two domains lo nemo Archaebacteria, Eubacteria untadi. These two are prokaryotes kada. Third domain lo undevevi Protista, fungi, plantae, animalia. Ivanni entivi eukaryotes kada. So ee two prokaryotes anamata, ivanni eukaryotes. That's why he called them as three domain system. I hope you got this. Okay, so thereby six kingdom classification ani dinki name ochindi. This was discovered, or this was classified by Carl Woese. You will learn about this system in detail at higher classes. Let us look at the five kingdom classification to understand the issue and considerations that influenced the classification system. Earlier classification system included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms, angiosperms under plantae. So ఇంతకు ముందు క్లాసిఫికేషన్లో బ్యాక్టీరియా అంటే ఫైవ్ కింగ్డమ్ క్లాసిఫికేషన్ కాకుండా ప్రీవియస్గా చేసిన క్లాసిఫికేషన్స్లో బ్యాక్టీరియా, బ్లూ గ్రీన్ ఆల్గే, ఫంగై, మోసెస్, ఫర్న్స్, జిమ్నోస్పర్మ్స్, యాంజియోస్పర్మ్స్ ఇవన్నీ ప్లాంట్స్లో ఇంక్లూడ్ చేసినారు. కానీ ఇప్పుడు ఆర్హెచ్ విట్టేకర్లో ఎన్ని ఇక్కడ ఇచ్చిన దానిలో ఎన్ని ప్లాంట్ కింగ్డమ్లో ఉన్నాయి? మోసెస్, ఫర్న్స్, జిమ్నోస్పర్మ్స్, యాంజియోస్పర్మ్స్ అంటే ఓన్లీ 4 ప్లాంటేలో ఉన్నాయి. ఫంగై వచ్చేసి ఫంగై కింగ్డమ్లో వెళ్ళిపోతుంది. బ్యాక్టీరియా, బ్లూ గ్రీన్ ఆల్గే వచ్చేసి మొనెరా కిందికి వస్తుంది. సో మొనెరా, ఫంగై, ప్లాంటే అన్నీ కిచడి పట్టేసి ఇంకా ప్లాంటేలో పెట్టేసినారనమాట. ద క్యారెక్టర్ that undefined the whole kingdom was all the organisms included has cell wall in their structure. అంటే ఈ మొత్తం క్లాసిఫికేషన్కి గ్రాస్ క్యారెక్టర్ ఏమంటే ప్రెసెన్స్ ఆఫ్ సెల్ వాల్. అన్నిటికి ఉంది కదా ఇక్కడ సెల్ వాల్. ఫంగైకి సెల్ వాల్ ఉంటుంది, బ్యాక్టీరియాకి సెల్ వాల్ ఉంటుంది, బ్లూ గ్రీన్ ఆల్గే కూడా సెల్ వాల్ ఉంటుంది. సో అలా సెల్ వాల్ ఉండేవన్నీ క్యారెక్టర్ చేసి ప్లాంటేలో వేసినారు. బట్ ఒరిజినల్లీ దట్ డస్ నాట్ బిలాంగ్ టు ప్లాంటే. సో this placed together a group which was widely different in other characters. So only cell wall lone they are similar, migitha annitlo they are totally opposite to each other. It brought together the prokaryotic bacteria. ade cheptunnam, bacteria, blue green algae are prokaryotes, fungi is eukaryote, mosses, ferns, gymnosperms kuda eukaryotes, and blue green algae cyanobacteria with other groups with eukaryotes also grouped with unicellular organisms and multicellular organisms, say ones. For example, ante ఇది కాకుండా ఇప్పుడు నెక్స్ట్ మనం ఇప్పుడు సెల్ వాల్ గురించి మాట్లాడినాం కదా, సెల్ వాల్ అన్ని సిమిలర్ ఉన్నాయి అందుకు ప్లాంటేలో వేసినట్టు. అది కాకుండా ఇప్పుడు యూనిసెల్యులారా, మల్టీసెల్యులారా అది చూసుకోకుండా వాళ్ళు ఏం చేశారు, క్లామిడోమోనస్, స్పైరోగైరా వర్ ప్లేస్డ్ టుగెదర్ అండర్ ఆల్గే. క్లామిడోమైనస్ వచ్చేసి యూనిసెల్యులర్, స్పైరోగైరా వచ్చేసి మల్టీసెల్యులర్ ఆర్ ప్లేస్ అండర్ ఆల్గే. దిస్ క్లాసిఫికేషన్ did not distinguish between heterotrophic group fungi. సో ఇలా ఈ హెటెరోట్రోపిక్ ఫంగై ఉంది కదా, ఫంగై ఈజ్ హెటెరోట్రోప్ అండ్ ప్లాంట్స్ ఆర్ ఆటోట్రోప్స్. ఆ ఫంగైని ప్లాంట్స్లో కలపడం ఇట్ వాస్ టోటలీ రాంగ్ బికాస్ ఇట్ ఈస్ నాట్ ఆటోట్రోప్, రైట్, ఫంగై. సో దట్ ఈస్ ఆల్సో డిసడ్వాంటేజ్. అండ్ ఆటోట్రోపిక్ గ్రీన్ ప్లాంట్స్ దట్ షోడ్ అ క్యారెక్టరిస్టిక్ డిఫరెన్స్ ఇన్ ద సెల్ వాల్ కాంపోజిషన్. మనకు తెలుసు ఫంగై యొక్క సెల్ వాల్ ఈజ్ మేడప్ ఆఫ్ కైటిన్, రైట్. ఫంగై సెల్ వాల్ ఈజ్ మేడప్ ఆఫ్ కైటిన్ అండ్ ప్లాంట్ సెల్ వాల్ ఈజ్ మేడప్ ఆఫ్ సెల్యులోజ్, రైట్. ప్లాంట్స్ సెల్ వాల్ ఈజ్ మేడప్ ఆఫ్ సెల్యులోజ్. And the plants wall is green plants, it is cellulosic cell wall. When such characters were considered, the fungi were placed in a separate kingdom fungi. ఎప్పుడు ఈ separate kingdom చేశారు? ఆర్హెచ్ విట్టేకర్ టైంలో కదా. ఏ prokaryotic organism were grouped together under kingdom Monera. సో ఇంకోసారి చెప్తాను, ఆల్ ద prokaryotic unicellularస్ని మొనెరాలో పెట్టారు, ఆల్ ద యూకారియోటిక్ యూనిసెల్యులార్స్ని ప్రొటిస్టాలో పెట్టినారు, ఆల్ ద మల్టీసెల్యులార్ యూకారియోటిక్ హెటెరోట్రోపిక్ ఆర్ సాప్రోఫైటిక్వి ఫంగైలో పెట్టినారు, ఆల్ ద మల్టీసెల్యులార్ ఆటోట్రోపిక్ photosynthetic organisms they are considered under plants, and all the with multicellular heterotrophic they doesn't have cell wall, they are kept or grouped under animalia. Okay, this is a very brief kind of classification of these kingdoms. Okay, now where am I? All prokaryotes are grouped under kingdom Monera and unicellular eukaryotes are placed under kingdom Protista. Kingdom Protista has brought together Chlamydomonas, chlorella, earlier placed in algae with plants. So chlorella, it is not a plant, it is a protistan. It is a protistan because it is unicellular and the cell wall is totally different composition and it is eukaryote. And earlier it was placed under algae in the plants, both having cell walls with paramecium and amoeba. So amoeba and paramecium were earlier placed in the animal kingdom, which they lack cell wall, but now they are placed in where? In kingdom Protista. I hope you are getting it. Okay, which were earlier placed in animal kingdom which lack cell wall. So you know that paramecium and amoeba lack cell wall. It has put together organisms which in earlier classification were put in different kingdoms. So ila konni changes అనేవి జరిగాయి. This changes will take place in future too. ఈ changes అనేవి ఫ్యూచర్లో కూడా జరుగుతాయి డిపెండింగ్ ఆన్ ద ఇంప్రూవ్మెంట్స్ ఇన్ అవర్ అండర్స్టాండింగ్ ఆఫ్ క్యారెక్టరిస్టిక్స్. సో మనం ఏ ఒపీనియన్, ఏ పాయింట్ ఆఫ్ వ్యూతో దాన్ని చూస్తామో దాన్ని బట్టి we will classify, right? So in that in future also there will be many classifications going on. And evolutionary relationships as well. Over time, an attempt has been made to evolve and classify systems which reflect not only morphological, physiological and reproductive similarities but also in the phylogenetical. Very, very, very important line. Over time, an attempt has been made to evolve a classification system which reflects not only morphology, means not only external characters but also the physiology, physiology and the structure, the way the outwards, the way the the composition, etc., and also reproductive similarities. How do they reproduce similarly, like a sexual or vegetative? But also in the phylogenic, in the sense of the DNA, the genetic material, etc., i.e. is based on the evolutionary relationships, how they are interlinked with their forefathers and etc. will also be considered under the classifications which would be done in the future. In this chapter, we will study characteristics of kingdom Monera, Protista, fungi of Whittaker classification. We will be dealing with Whittaker's classification. The kingdom Plantae Animalia are commonly referred to as plants and animals, which we know. Respectively, we will be so we will deal with them separately in plant and animal kingdoms in the third and fourth chapter. Okay, now let's jump into the kingdom Monera. Okay, kingdom Monera. Bacteria are the sole members of kingdom Monera. Kingdom Monera lo manamu మొత్తానికి చదివేది ఒక్కటే ఒక దాని గురించి, which is bacteria. All the bacteria concentration is much in the Monera. Monera, in the sense, synonym of Monera, it is bacteria. Okay, you remember in that word, that way. They are most abundant microorganisms. What are the most abundant microorganisms? They are not viruses. Viruses are not considered as living. If we consider viruses as living, then you can say viruses are the most abundant. But besides viruses, the most abundant are bacteria. Bacteria occur almost everywhere. It means they are cosmopolitan in distribution. Everywhere, in the sense they are present in 100, more than much high temperature. They are present in freezing, or they are at the lowest temperature they are present, at the highest temperature they are present. Extreme conditions lo untayi anamata bacteria. Hundreds of bacteria are present in a handful of soil. So this line is also very, very important. Hundreds of bacteria are present in a handful of soil. They also live in extreme habitats such as hot springs, deserts, snow and deep sea oceans where very few other life forms can survive. So వీటితో వేరే ఆర్గానిజమ్స్తో కంపేర్ చేస్తే అవి బతకలేని చోట బ్యాక్టీరియా బతుకుతాయి. అంటే దే ఆర్ వెరీ స్ట్రాంగ్, దే ఆర్ వెరీ రెసిసివ్ టు ఎక్స్టర్నల్ స్ట్రెసెస్ అనమాట. దే హ్యావ్ వెరీ we will learn in future about this bacteria in detail. But for now we will be only discussing introduction part. So చాలా అంటే చాలా స్ట్రాంగ్గా ఉంటాయి దాని సెల్ వాల్ కాంపోజిషన్. సో that is the reason why they are able to survive in these much extreme conditions అనమాట. So bacteria are grouped under 4 categories based on their shape. So dani external shape prakaram they are considered under 4 categories. They are spherical cocci, rod shaped bacillus and the comma shaped vibrio. Spherical ante manaku telusu, they are round shaped. Here you can see these are cocci, and bacilli are rod shaped, so these are spores. And this spiral shaped or spring shaped are known as spiral or spirillum. And vibrio are comma shaped. These are comma shaped, they are known as vibrio. So this is the classification of bacteria based on their shape or morphology. Now, though the bacteria structure is very simple, సో చూడడానికి అవి చాలా సింపుల్గా ఉన్నా కానీ దే ఆర్ వెరీ కాంప్లెక్స్ ఇన్ దర్ బిహేవియర్. వాటి బిహేవియర్ ప్రకారం చూస్తే దే ఆర్ వెరీ కాంప్లెక్స్ విత్ దే షో వెరీ కాంప్లెక్సిటీ ఇన్ దర్ బిహేవియర్ బట్ దే హ్యావ్ వెరీ సింపుల్ స్ట్రక్చర్. కంపేర్డ్ టు మెనీ ఆర్గానిజమ్స్, bacteria as a group show the most extensive మెటబాలిక్ డైవర్సిటీ. సో దాని మెటబాలిజం అనేది మోస్ట్ ఎక్స్టెన్సివ్, కాంప్లెక్స్, డైవర్స్ ఉంటుంది అనమాట. సమ్ ఆఫ్ ద బ్యాక్టీరియా ఆర్ ఆటోట్రోప్స్. కొన్ని బ్యాక్టీరియా మాత్రమే ఆటోట్రోప్స్. దే సింథసైజ్ their own food from inorganic nutrients and they convert them into ATP and they perform various biological reactions. Okay, but vast majority of the bacteria are heterotrophs. So కొన్ని మాత్రమే ఆటోట్రోప్స్ ఉంటాయి. Majority of them are heterotrophs. They depend on other organisms or dead organic matter for food. So those are known as heterotrophs. So the question may arise, what majority of the bacteria are? Options will be saprophytes, parasites, heterotrophs, autotrophs. The correct option is yes, is heterotrophs. And you may ask me, ma'am, sorry ma'am, sister, whatever. So you told me that these are heterotrophs, but you are telling parasite, saprophyte, they are also branched under heterotrophs. But NCERT line to line questions will be asked. So you should be stick on, you should be mad, and you should by heart the line that they are heterotrophs. That's it, gun shot. Okay, means they will be not asking like that what I have asked, but sometimes it just happens. Next, first we will be dealing with the first type of bacteria, which are archaebacteria. Archaebacteria, it is commonly known as primitive bacteria, and archaebacteria are also known as primitive and also ancient bacteria as well. So the cell wall is consisting pseudomurine. It is extra information, if you want you can note it down. So branched chains lipids are present. That is why they show very complex cell wall structure, which makes them sustain or live in very harsh climatic conditions. These bacteria are special since they live in some of the most harsh habitats, such as extreme salt areas, halophils, hot springs, thermoacidophils, and marshy areas, methanogens. So those which live in extreme salt areas, ఎక్కువ సాల్ట్ కాన్సన్ట్రేషన్ ఉండే చోట ఉండే దానిలను హాలోఫిల్స్ అంటాము. ఎక్కువ వేడిగా ఉండే వాటర్స్లో ఉండే దానిలను థర్మోసిడోఫిల్స్ అంటాము. ఎక్కువ బయోగ్యాస్, మీథేన్ ప్రొడ్యూసింగ్ ఏరియాస్లో ఉండే దానిలను మెథనోజన్స్ అంటాము. Archaebacteria differ from other bacteria having different cell wall structure. Pseudomurine is the component which is present in its cell wall, and the cell wall is cell membrane is branched chain lipids, and that is what makes the cell wall very special, and it helps in sustaining in very harsh climatic conditions. That is the reason. And methanogens are present in the gut of several ruminant animals. Ruminant animals are such as cows and buffaloes. Ruminant animals యొక్క gut అంటే స్టమక్లో ఈ మెథనోజన్స్ అనేవి ఉంటాయి. సో దట్ ఈస్ వై యూ కెన్ నో దట్ డంగ్ని మనం డీకంపోజ్ చేస్తే we will get biogas, right? That's the procedure behind this. Are responsible for the production of biogas methane from the dung of the animals. Can anybody write the formula of methane in the description box? Okay, I'll be waiting for the answer. Anybody? Next, eubacteria. There are thousands of different eubacteria or true bacteria. So this is another classification of bacteria. First one is archaebacteria. Next one is eubacteria. Eubacteria, it is also known as true bacteria. Eubacteria, thousands of eubacteria are there, but we will be dealing only few. Okay, there are thousands of different of eubacteria, true bacteria. They are characterized by the presence of rigid cell wall. Here tough cell wall is there, but here rigid cell wall. And if motile, if the bacteria, if it is motile, flagella is present. If it is not motile, flagella is absent. The cyanobacteria, it is a type of eubacteria, also referred as blue-green algae, but it is not an algae. Eubacteria, it is also called as blue-green algae, but it is not a algae. It contains chlorophyll A, similar to higher plants. You know that one thing, in the higher plants we will be having chlorophyll A and chlorophyll B. These two pigments are present in the higher plants, but chlorophyll A, it is seen in blue-green algae. So that is why it is somewhat linked with the higher plants, which is mentioned in the NCERT. You can see here, right? These are containing chlorophyll A, similar to green plants, and are photosynthetic autotrophs. Blue-green algae or autotrophs, obviously by that name you can remember that these are photosynthetic autotrophs. The cyanobacteria are unicellular, colonial, or filamentous. So here, based on the cell number, they are classified into either unicellular or colonial or filamentous. Freshwater, they can either live in freshwater or marine water or salt water or on terrestrial algae as well. The colonies are generally surrounded by gelatinous sheet. Gelatinous sheet in the sense a sticky substance which is attached on the surface of this nostoc. You can see the blue color highlighted outer surface. It is a filamentous blue-green algae. So this is the gelatinous sheet which is surrounded along the colonies, so that the cells will be combined and they will be merged together. They often form blooms in the polluted water. So these are good indicators of pollutions. So they form algal blooms on the polluted water. Some of these cyanobacteria, they can also fix the atmospheric nitrogen in the special cells known as heterocyst. This labeling is very important. They can be asked for the MCQs as well. This is heterocyst and this is mucilaginous sheet, and this is nostoc. Very important. Sometimes direct question of this labeling will be asked, or they can give the picture and they can ask that what is this picture, what is the organism present in this picture. So these are heterocyst, which you can see here, violet color thing, and this is nostoc. And anabina can also fix the nitrogen. And chemosynthetic autotrophs. So we have seen a photosynthetic autotrophs, like, right? And now we will see chemosynthetic autotrophs. Now you can get a confusion. What is it chemosynthetic? What is this photosynthetic autotrophic nutrition? Turn the page again. Come to the Bible, whatever, for the biological classification. I am saying that this is the Bible, this is the Quran, and this is the Bhagavad Gita, whatever it is for the biological classification. Monera, you can see, right? These are prokaryotes. What is prokaryote? Which does not have well-defined nucleus and well-defined, which does not have cell membrane bounded cell organelles are absent. Those are known as prokaryotes. Cell wall is non-cellulosic, and cell wall is made up of polysaccharides and amino acids, and nuclear membrane is absent. This is very important. Body organization is cellular. It means they are unicellular in nature. And mode of nutrition, very important column, you should remember this. Autotrophs, it means they can prepare their own food. Autotroph, this you can get confusion. Autotrophs, they are divided into two types: chemosynthetates and photosynthetates. Chemosynthetates, they are deriving the nutrition from chemicals. So chemosynthetates and photosynthetates. Photosynthetates, they derive their nutrition from sun, but chemosynthetates, they derive the nutrition from chemicals. And next, heterotrophs. Next, heterotrophs are again of two types. You can see here, saprophytes and parasites. Saprophytes and parasites. So this is the brief division of the kingdom Monera mode of nutrition. If you get this, I am damn sure that you will have a good catch of the subject. Now, eubacteria, chemosynthetic. It means still up to now we have completed photosynthetic bacteria. Now we will be dealing with the chemosynthetic autotrophs. Bacteria oxidizes various inorganic substances such as nitrites, ammonia, and use the released energy of further ATP production. So in the photosynthetic autotrophs, they use the sunlight, but here they use the inorganic chemicals such as nitrites and ammonia, and from them they derive the nutrition, and nutrition in the sense the energy required for the ATP synthesis, and they play a very important role in recycling the nutrients. This chemosynthetic autotrophs, they are very crucial for recycling nutrients like nitrogen, phosphorus, iron, and sulfur. So these element names are very important, and they may be asked as questions, which are nitrogen, phosphorus, iron, and sulfur. These are very important. And next is heterotrophic bacteria. We have completed photosynthetic, chemosynthetic. Now we will be dealing with heterotrophic bacteria. Heterotrophic bacteria are most abundant, which we have known. Minority of them are photosynthetic, but majority of them are heterotrophic. Heterotrophic are the most abundant in the nature. The majority of them are important decomposers. So in that heterotrophic also, majority of them are decomposers. Majority of them are decomposers. So that is what it is given here, that majority of them are important decomposers. Many of them have significant impact on human affairs. So many of them chala mana human beings ki chala effect chupistayi anamata in our daily life. They play a very important role on humans life as well. They are helpful. So first vachesi lactobacillus, streptococci, these are all bacteria which are present in the cud, and they help in turning milk into curd. Important thing which is essential for converting milk into curd. It is also bacteria. And production of antibiotics, antibiotic production few are very important in producing antibiotics as well. Fixing nitrogen in leguminous plants, in the leguminous roots, example, etc. Some are pathogenic, causing damage to the human being, crops or farms, animals and pests. Some are pathogenic. So in heterotrophic we can see some, right? We have seen in heterotrophs they are maybe saprophytic or parasitic. Some are saprophytes and some are pathogens, are they cause diseases, damage to human beings or crops or farm or animals or pests or pets. Not pest, it is pets. There are some diseases which are caused, cholera, typhoid, tetanus, citric cancer are well-known diseases caused by different bacteria. So cholera, typhoid, tetanus, citric cancer are 4 types of diseases mentioned in NCERT that are caused by the bacteria. Very important, just remember them, they are very very important. And now let's go into the reproduction which is shown by bacteria. Bacteria reproduces mainly by fission. Now what is fission? Fission in the sense, it may be binary fission or fission. Mainly by fission. Now what is binary fission? You all know binary fission in the sense, if this is a వస్తున్నా 5 minutes. ఇది ఒక బ్యాక్టీరియా అనుకో. This is another if this is a bacteria, first bacteria reproduces mainly by fission. Now sometimes under unfavorable conditions they produce spores. So during some unfavorable conditions, this bacteria mainly reproduce by fission. So binary fission is nothing but, in a short form they call it as fission. Sometimes under unfavorable conditions they reproduce by spore formation. Now what are spores? Spores are some fruiting bodies which will in turn give the full body of the bacteria. It is an asexual reproduction. They also reproduce by sort of sexual reproduction by adopting primitive type of DNA transfer from one bacterium to another bacteria. And primitive DNA transfer, it is an old type of DNA transferring shown by the bacteria. That it is given here, a dividing bacterium. So first the cytoplasm will be merged and then the DNA will be divided and it will be coming close and it will stick together and DNA transferring nucleus transfer will happen, and then the all will be coming close and giving a new daughter cell kind of a thing. That's how sexual reproduction happens in bacteria. Here this is very important, labeling is very 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 important. This is cell wall, this is cell membrane. Green color outward line is cell wall, inner line is cell membrane. This structure which is given inside, right, these two are DNA. Now mycoplasma, the mycoplasma are the organisms that completely lack cell wall. Now we have seen right here in our Bible or in our biological Bible, here classification ka Bible, okay, whatever. So in Monera we have seen the cell wall is non-cellulosic, not with made up of cellulose but with polysaccharides or amino acids, right? So that on that point of view we would be imagining that like everything in this Monera have cell wall, they have cell wall, they have what you people will be doing means yes, Monera contains cell wall which is non-cellulosic which is either made up of polysaccharides or amino acids. You will be batting it, but that's not true. Some are exceptions which are mycoplasma, which we will be dealing very soon. The mycoplasma are organisms, I have just made a fun, that's not, but yeah, few people will be there who will be not having they will be not even knowing what they are reading, but they will be just by hearting everything. Mug-upping is not final option, but you need a little, I know at some point of view you have to mug up some concepts, but few things you can add some basic sense and you can remember. Mycoplasma are the organisms that completely lack cell wall. They are acellular, they don't have cell wall. They are smallest living known creatures. So you can have again a argument that why you are telling mycoplasma is the smallest. Viruses are even more smaller, right? But they are acellular, they are non-living outside the host, right? So they are not completely living. So we should not consider living those viruses as living. So keeping that aside, these can live without oxygen. Means they are known as facultative anaerobes. They can live without oxygen, without supply of oxygen. Mycoplasma, another name is jokers of plant kingdom. You can note it down. And many mycoplasma are pathogenic in nature, and in plants and animals they cause some diseases, this mycoplasma. That's what given in the kingdom Monera. I hope you got this well. If you really understand, thank you. And I'll be soon uploading kingdom Protista fungi as well. We will soon complete this chapter, and I'm sure that I'll complete first chapter as well very soon. And if you like the explanation, like, share, subscribe for new videos. Hope you give a good support, and we will be making a good team in future. Thank you for watching. See you next video. Bye.